Hello, everyone. So we're gonna probably talk a little bit more about the contraction cycle and then also about how the nervous system works today. So as captured last time, our contractile organelle is a muscle fiber. Muscle fiber. Within a muscle fiber, it's called a myofibril. You have tons of these myofibrils contained within one muscle fiber, like so. And then one myofibril contains, if you zoom in, a lot of myofilaments. So myofilaments and then myofibril. And the myofilaments can be either thick or thin filaments as appropriate. So what's the implication of this is that these myofiber filaments in your myofibrils with these thick and thin filaments are organized in repeating units called sarcomeres. So a sarcomere, they essentially look like this, but one unit like this, and then they're tethered together. So when you're contracting the muscle, the myofilament, the sarcomeres will move inwards and contract. And basically they're going to form these actin-myosin cross bridges here. Cross bridges here, which create tension inside the muscle. So let's label some parts of this. So this middle part, it's going to call the M line, and this part is going to be called the A band. A is because it has all. These are going to be thin filaments here. And so these are going to be called Z bands, what we see here. Z bands. Same thing here, Z bands. And with each Z band, you're going to have an I, or Z line, you're going to have an I band here. Because you have thick filaments here, whereas you have thin filaments. I should label thin and thick. So the I bands here are the ones with exclusively thin filaments, and they're going to have this a sort of overlapping space. These thin filaments are made of actin. So thin, let's say, is actin and other sorts of proteins. But the big idea is you can have actinine, actin, and stuff like this. If you're ever asked to label, the big idea is you have actin, you have actinin, you have tropomyosin, and you have troponin. So I can explain each of these a bit later, but the idea is if you ever have like a drawing of how it's like formed, something like this, we'll have something like this. The middle line here is called nebulin. And then these big blocks, these red blocks, are going to be called actin. Tropomyosin is going to be the blue line here, tropomyosin, and then myosin, and then troponin is here. Is here. Actinin is going to be forming the Z lines. So they're going to be forming, interconnecting the thin filaments like here. So the thick filaments are much simpler. They have these sort of thick heads. So if you go down here, they're just going to look like these, and they're going to have these sort of myosin heads built like this. And at the same time, on all these filaments, they're kind of attached to something called titin, which connects to Z line. Z line being this part here. You have the myosins here. So in a specific skeletal muscle contraction, meaning when your muscles do action, move, and do whatnot, what generally happens is that first, let's see, you're going to contract after you get calcium levels increase. So remember how calcium is increased is first you got to have the NMJ, you have the motor neuron setting acetylcholine entering through the NMJ. And then upon entering the NMJ, it's going to trigger an action potential from the T tubules. And then this action potential is going to be brought to the sarcoplasmic reticulum. And then the SR is going to release calcium into the muscle. And then after calcium levels increase in the muscle, you will start to contract. As long as these become elevated. What calcium does is they interfere this tropomyosin complex. They bind to troponin and reveal myosin binding sites on actin. So this occurs with a cycle. Step 0, so I'll list them in steps of the muscle contraction. So step 0 is when calcium is released, and then calcium reveals binding sites. So if you want to draw it out, you can see if this is, say, myosin. So calcium is binding here. This is step 0. Step 1 will be after it's bound. Well, I should label. This myosin head has ADP plus P. After it's bound, myosin is going to bind onto actin, and then form cross bridges. Form cross bridges. Form cross bridges. And then it's going to move. And then it's going to pivot. Pivot. So I'm going to draw like it pivoting. And then upon this pivot, it's going to release ADP plus P. Well, this is step 2, I should say. It's going to pivot and it's going to slide. And then step 3. Well, step 3 is ADP being released, I should say. It's a step. And then step 4, I should say, is that after moving, ATP is going to bind onto this complex. And then the myosin is going to detach. And after step 5, ATP is going to be turned into ADP plus P, and then it resets. The cycle resets itself. So that's kind of the gist you got to know. You got to know that it starts with this ADP plus P complex. Calcium reveals the actin binding site for myosin. And then upon attaching, the myosin heads release ADP plus P upon making cross bridge, I should say, it removes ADP plus P for the power stroke, for the movement part. And then afterwards, you apply ATP to reset the cycle, to detach it, and then the hydrolysis movement kind of just resets the entire thing. So let's see what else to do. By contracting, you're going to get more tension. You have more tension. And tension is based on the number of overlapping myosin actin interactions. So if you have more myosin actin poles, you have more tension being produced. And maximum tension is at intermediate length. This is because you have the greatest number of cross bridges formed. And calcium is also an important role regarding how many you have, I should say, like how much calcium you have. Now, you can also have repeated summation. So first you can imagine a twitch. This is when calcium levels are increasing, and this is when calcium levels are decreasing. And this is one twitch. Now, the case is you can actually have summation. If you repeatedly stimulate a muscle fiber, you can have more calcium buildup. So you can have like it's building up now, and then again and again, and then again, and then you can rest here. So summation is when summation, more calcium enters before original leaves. This leads to more active sites revealed. One case is you can get tetanus, actually. In the case of tetanus is when you have persistent tension. You have persistent tension produced by stimulation. And when this happens, you're gonna just get a stressed out muscle, and you're gonna just always get a sort of, I don't want to say damage, but you're gonna just be very tensed up. Maximum tetanus. So yeah, let me see. Uh, there's also forms of concentric, there's also forms of movement such as concentric, isometric, and yeah, eccentric. So let's just quickly write that out. Concentric is when the tension built in the muscle, the external load, I should say, load is less than the tension, uh, leading to movement of a joint. This is like kind of like if you're lifting something, then the external load is less than your internally generated tension. So you're able to shorten the muscle and you're able to make something happen. And the other is isometric, is load is equal to the tension of the muscle, and you're just holding it. So imagine, let's say you're hanging off a cliff or something, and you're gonna feel that there's tension in your muscle, but the load and whatnot in your actual perceived tension is gonna be around the same. And so nothing really changes in that case. The length doesn't change is the biggest case. And the other one is eccentric. This is when the load can be greater than the tension produced. And this is like, let's say you're doing stretches or something. This could mean that the muscle is getting longer rather than shorter and can be used kind of like a brake to kind of slow down what's happening. Now let's talk about force and work and just some sort of energetics and stuff. Muscles can get ATP from using either aerobic or anaerobic sources. So aerobic means using O2, and then anaerobic means no O2. Aerobic happens in the mitochondria, whereas anaerobic happens in the cytosol. Now, the biggest difference is that aerobic is slow, can also use fatty acids, but is efficient. It produces 30 ATP per one glucose, whereas anaerobic is fast, but is inefficient, only producing 2 ATP per glucose, and you cannot use fatty acids. There's also another one, phosphocreatine. You don't need O2. It's fast, but it's limited in storage, meaning it depletes really quickly. So whether or not your muscles are working out or exercising is going to have a lot of differences between how they obtain ATP. If you have a muscle that's resting, you're going to have use of fatty acids, reduce ATP surplus, and you're going to build up glycogen and creatine phosphate stores. If you're moderately active, you use up your fatty acids and glucose and use your glycogen stores. Now, if you're at your peak performance, you're going to use everything. You're going to use glucose, fatty acids, and also creatine phosphate, and waste products, which includes lactate and creatine, are going to build up in your muscles. This is going to show fatigue essentially. Um, yeah. So let me see what's up with this. Well, I should say when you're peak activity, you're going to be not using anaerobic. Well, okay, the key thing is this is using aerobic. These are using aerobic, but this is using anaerobic. Aerobic plus anaerobic. So you would only really use anaerobic respiration when you are doing peak activity in this case. So if you're ever given a question where you see the use of anaerobic respiration, it might be a clue. That is, it's being peak activity at the moment. So fatigue occurs when your ATP production is unable to keep up with the contraction cycle or your movement or whatever you're going to do. So there's going to be two types of factors. You can have factors affecting excitation, and you're also going to have factors affecting contraction. In terms of excitation, you can have accumulation of potassium in T tubules because you need to move stuff. So potassium, if you have too much potassium, it means that you definitely need to reset a bit. And no more AC acetylcholine vesicles in motor neuron subterminal. So you're being depleted of acetylcholine vesicles. You can't move acetylcholine. And then you can have micro tears in terms of contraction in myofibrils. And you can also have a build up of lactate and hydrogen, and also have leakage of calcium back into sarcoplasm. In short-term exercise, one thing you can do is lactate can be shipped to the liver for gluconeogenesis, making more glucose. But in medium or long term, myosatellite cells use amino acids to fix torn myofibrils. Now, importantly, there's a trade-off you kind of have to imagine. Your bodies are going to be different if you're a long distance runner versus if you're a weightlifter. You have a difference between whether you want your muscles to be quickly fatigued or able to do a lot of power in a short amount of time. So this is why we have distinct types due to timing, protein composition, and organelles. So you have type 1 muscles, which are slow oxidative fibers, and you have fewer myofibrils, but more mitochondria per fibril. Slow myosin, slow pumps, and more capillaries. And then you have type 2, which splits into type 2A and type 2B. Type 2s are all known as fast twitch, and they all have more myofibrils, less mitochondria, fewer capillaries. And A is known as intermediate, and B is known as fast type 2 fibers. So I'll go first with the fast. Fast are known for being for basically having fast contraction speeds and whatnot. Whereas this is more of a medium in terms of mitochondria number, capillary density, recovery and resistance, and also power. So you have to remember at the same time, muscle fibers are split into motor units, which means it's one motor neuron attached to one myofibril, myofiber, I should say, right? And because it works like this, one motor unit's activation is all or none, which means that, well, first thing is that all the fibers in a motor unit are of the same fiber type. And this means that because it's all or none, you have this effect that happens where you're going to use probably the weakest or the least amount of recruitment that you need for myofibers to move something rather than use the greatest amount of force. You're not going to tense your entire arm to lift up a pencil, for example. So let's see what's a good example of this. Mm. So small motor units, I guess you can say. Small motor units are few fibers with all being type 1, whereas largest are going to be many motor units with all being type 2. And the recruitment is only done such that there's a size principle. So only smallest motor units recruited first for stimuli. So what you're going to see is first you're going to see type 1s, smaller ones, and type 2s, and then you're going to see type 3s. So you're going to see this sort of hierarchy. Mm. And muscles can enlarge their peak tension production with two ways, hyperplasia and hypertrophy. Hypertrophy is increasing the number of myofibrils in per myofiber, and hypoplasia is the number of myofibers per fascicle. And humans can only use hypertrophy, only through the increasing the number of myofibrils. Although you can have some sort of conversion between the sort of muscle types that we mentioned, whether it's from type 1 or type 2 or on and on, right? So I digress. Let's go to the nervous system. Nervous system is made out of three components. The central nervous system, which is your uh I should say your brain and spinal cord. Your peripheral nervous system, which is everything else basically, and your enteric nervous system, which is neural cells in digestive system, which connects directly to the CNS. Uh, so yeah, as I mentioned, the CNS is composed of the brain and spinal cord, as the PNS is made out of nerves and ganglia. And yeah, I guess there's three types of classifications. You can have sensory neurons, you can have interneurons, and you can have motor neurons. So sensory is like if you detect something, it's your sensory neurons are going to pick up onto it first, and you're going to pass information from the outside environment into the nerve system. Interneurons pass information between neurons, while motor neurons pass information to non-neuronal cells. Now, information transfer uses electrical signals and chemical signals. This can be in the form of acetylcholine or in the form of potentials. Importantly, we have two types of cells. One type are neurons. This is a primary basis of the nervous system, and the other form is neuroglia, which has supporting roles. you have the site, the shape of a neuron, axon, dendrites, axon terminals, and whatnot. And you have to know that information is unidirectional. There's also many ways of classifying neurons. You can have, say, a neuron is like this. We have multiple dendrites. You have multiple dendrites in one axon. This is going to be called multipolar. If you have an axon with two dendrites, this is going to be bipolar. If it's going to be pseudo-unipolar, that means it's going to be one axon paired to a soma, then dendrites composed to an axon. This kind of extra information. Pseudo-unipolar is basically you have one axon connected to a soma, and the dendrites connect directly to the axon. And then you have an anaxomic, which kind of looks like a sun, which is basically composed of many neurites that act both as dendrites and axon terminals. There are many ways to classify neurons and whatnot, but you can understand that neurons can be classified as being part of the somatic nervous system or the visceral or the autonomic nervous system. So autonomic nervous system is kind of the same as motor is composed of motor neurons that are tissues other than skeletal muscle, whereas somatic is more of just motor neurons innervating skeletal muscle. Let's talk about glial cells first, or neuroglia. You can have ependymal cells, microglia, myelinating cells, and then astrocytes and satellite cells. So ependymal cells have different, and microglial cells have different function. Ependymal cells lines the ventricles and secretes CSV. And then microglial cells are white blood cells for immune functions, which modify connections. Myelinating cells produce myelin in both the CNS and the PNS. In the CNS, it's going to be called oligodendrocytes, whereas in the PNS, it's going to be called Schwann cells. And myelin allows for efficient and rapid signal transfers. Astrocytes and satellite cells have diverse functions. Most importantly, they can regulate CSF flow. Oh, also, astrocytes are in the CNS, whereas satellite cells are in the PNS. One thing you can imagine is that the brain is, I guess, the astro like a star, and then everything surrounding it, the PNS, is going to be satellite cells, which kind of makes sense that way. So this is more of a CNS function and regulate CFS flow, and then recycle neurotransmitters. This is both CNS and PNS, but this is more PNS, I'd say. And then you can buffer ion concentrations in extracellular fluid. This is more PNS. You can also regulate blood flow, which is more PNS action. Neurons can't be born in adulthood, unfortunately. So there's no such thing as neuron therapy, neuron transplants, or anything of the sort. But what can happen? So when it dies, it can't really be replaced. And how they die? They can have physical damage. You can have excitotoxicity, which is increasing excitatory activity. And you can also severe axon. You can sever the axon. And if it's in the PNS, it can actually heal with glial cells. Schwann cells can help it heal. Schwann cells heal in PNS. But if in the axon cell, if in the CNS, astrocytes inhibit axon healing. This is something key. So astrocytes are the ones that make it from not working. They're also composed of different sorts of tissues. In the CNS, you can be divided into both white and gray matter. So white matter is usually involved in voluntary, well, white matter is myelinated axons, whereas gray matter is unmyelinated. And then, so white matter is related to awareness and voluntary control, whereas gray matter is more related to reflexes in terms of circuits. Okay, so let's draw this out actually. In your spinal cord, when you relay a sensation, it first goes through a dorsal gray matter here. And then you have, well, it's going to look kind of like this. And then you have dorsal gray matter, ventral gray matter, which is a motor neuron, sensory processing. And then you have dorsal white matter going to the brain, which is dorsal white matter, sensory awareness. And then you can also have lateral white matter going down, which is voluntary control. So the idea is white matter is dealt with kind of more of active control, whether you want something to move, you want something to do something in your body, whereas gray matter is more of just like reflexes. This is why you'll see like people who are handicapped, who might be in wheelchairs and stuff, they are they are able to essentially what's it called? The They are still able to have reflexes. Uh, Sorry, give me a second. They're still able to have reflexes, but they aren't able to, let's say, move their leg by command and stuff, or move their arm by command. So, okay. What we should also be aware of is the case of membrane potentials, graded potentials, and how we're going to get these sort of specific signals, I suppose. And that's also something we should consider. So, all cells have a transmembrane potential. Let's say we have a cell like here, membrane potential. So this means if we were to stick something here to measure the potential, you can see a charge generally in the cell. Uh, and when an excitable cell is excited, it's going to be less negative. It's going to be more positive. So more positive is going to be excited. And excitable cells, they can alter their membrane potential, transmembrane potentials to send or receive signals. This can either be in a graded potential or this can be in an action potential. Now, these signals occur by proteins that allow ions to rapidly cross the plasma membrane. So I guess the idea is what is building up these membrane potentials? What kind of helps and what is an action potential? I guess these are the questions that we're going to ask. So the first thing is that we have what's called the resting membrane potential, which is around -70 millivolts. So potentials are defined as changes from the resting membrane potential. So this is going to be depolarization, and the repolarization is going down, and you have hyperpolarization, which is going super negative. This is what an action potential looks like. So the potentials, all potentials are created through leak channels for the resting membrane potential. Well, they're all created through ion channels, but gated channels can lead to action potentials and graded potentials. This can include ligand gating, voltage gating, or mechanical gating. The resting membrane potential is due to an unequal permeability of ions. This is due to differences in leak channel abundances. So what does this mean? Uh, let's say potassium was the only permeable ion, and there's much more potassium inside of the cell than outside. This means potassium is going to move from the inside of the cell to outside of the cell. But the second this starts moving, well, we know that potassium is positive. So initially this place is really positive, but let's say it starts moving out here. That means the outside of the cell starts being more and more positive. And because it becomes more and more positive, it creates a net negative charge inside the cell. And this leads to some potassium going back. And then net movement ceases when opposing forces are equal. This is what's going to be called the equilibrium potential. Also known as what's it called? The transmembrane potential. Transmembrane potential when net movement is zero. There's still movement, just the net movement is zero. And so these are kind of ideas that could be should be understood. In the body, there's Na and K gradients maintained by the NAK ATPase pump. And you also have Cl gradients using co-transporters with K. These are ways to do it. Ion pumps, they only have a small effect on resting membrane potential. It's more as though the leak channels that maintain resting membrane potential, the things that are allowing it to move left and right easily. And signals, potentials, like action potentials and graded potentials are moved by using gated ion channels. Uh, so signals in an excited membrane are driven by the opening and closing of these sort of gated ion channels. And gated ion channels are just, yeah, just like that. The size of a graded potential is also variable. It's affected by the number of ion channels that are open. So more ion channels open, more potential, more permeability. Graded potentials can last for tens of milliseconds and have different polarities, meaning directions, as well as amplitudes, meaning how much they do. And graded membrane potentials are generally they spread from the original sites, but they get weaker and weaker. So you want something that can essentially find a way to stretch across your body. Because let's say your brain needs to get a signal all the way to your leg. That's really tricky. So they're too long to rely on graded potentials for transmitting information. So you need an action potential. Action potential, which is a specialized signal that can be transmitted through long distances. So key characteristics are they have an all or none threshold. They have propagation, stereotyped dimensions, and they are and they have a refractory element. So you should have an all or none threshold. All should be refractory. Some similarities, though, are that they both use graded potentials And yeah, it's they just are generated using gated potentials. Uh, Action potentials are generated by opening and closing two types of voltage-gated ion channels. The shape depends on the opening and closing of both. So initially, the voltage-gated sodium channels, let's just draw it out. So initially, voltage-gated sodium channels open rapidly and then close. So let's say it's here. So Na open until threshold, and then Na close. And then the sodium and activation gates are closed. And then voltage-gated potassium channels are starting to open, and then potassium channels close here. But potassium channels like open very slowly and also close very slowly. So they have to react kind of through a delay. That's why we see this hyperpolarization period. And these kinetics lead to a refractory period. So essentially, if you were to draw it out again, there is a period here. This is called the absolute refractory period. This is because during this time, a second AP during this time, a second AP cannot be triggered no matter how strong the stimulus is. Well, it actually should be more accurately to be here. This is because the Na gates are all closed at the moment. However, during the relative refractory period, a larger than normal stimuli can trigger another a action potential. Well, there's a specific reason for this. Uh, yeah, it's because during this time, yes, it's as I said, during this time specifically, during the absolute refractory period, the Na channels will be closed. Whereas in the relative refractory period, there's a time when it's open. Uh, Myelin, you should understand myelin increases conduction speed and insulation for the action potential, and they propagate themselves by replicating them again at another part of the action potential. Well, let me see if I'm missing anything. Nope. Okay, now after getting into the synapse, what will happen then? This is a question that we might also ask. Well, upon making it to the synapse, you get the circuit, you have the AP, synapse, another AP. You have a presynaptic area cleft, and you also have a postsynaptic cleft. And these sort of axon terminals, sorry, my slides aren't working very well right now. These axon terminals are compartments that release neurotransmitters in response to an action potential. Upon receiving this action potential, there's going to be a voltage-gated calcium channel. And these voltage-gated calcium channel converts AP, depolarization, into release of calcium. Uh, and this is done through, well, as we mentioned, through acetylcholine, where you have the action potential being transmitted through T tubules, and the T tubules, well, no, the action potential is in the motor neuron, and the motor neuron secretes acetylcholine, which acetylcholine converts into another action potential inside the muscle, which goes into the T tubules and secretes calcium. Now, neurotransmitters released into the synaptic cleft bind into it. And I should say, I guess since we're getting into it more specifically, acetylcholine binds into the synaptic cleft, and this releases sodium, which triggers an action potential. So one action potential triggers another, and this action potential, the secondary action potential, this release calcium release. So you should remember that there are also many different types of ion channels that we discussed earlier. One's idea is we have ligand-gated ion channels, which can receive a neurotransmitter and produce a postsynaptic potential, which is what we're seeing here. Would be a ligand-gated ion channel, and the postsynaptic potential is here. And the postsynaptic potentials can be either excitatory or inhibitory. Excitatory keeps towards the transmembrane potential towards the AP threshold, whereas they keep it away from the AP threshold. Inhibitory keep it away from the AP threshold. And it's definitely, again, amplitude is affected by the number of receptors available and the number of ligands available. So if you increase the number of ligands and increase the number of receptors, you'll see a much greater potential and more ions can pass. Two of these potentials can summate together, or they come across the same patch. So let's say you have it here, here, and they add together and make a third one. So you can have one, the second one, and 1 plus 2 is going to be equal to 3. And this is going to occur in two different ways. Let's imagine we have a axon or whatnot, and you have two different innervations here. You can have, if they're from two different axons, then this is called spatial summation. Two different. But if it's just from one that's secreting two at the same time, this is called temporal summation, which is consecutive PSPs by the same axon. Now, more specifically, neurotransmitter receptors, they aren't all ion channels. You can have metabotrophic that can affect biochemical signaling, and you can also have ionotrophic, and we'll probably discuss this later. Probably not that much. You should know that an agonist activates a receptor in the nervous system, and an antagonist will they block a receptor. All these synaptic vesicles and stuff are recycled in the axon terminal. So this is vesicles and neurotransmitters are recycled in the axon terminal. And this is done through many ways. You can have acetylcholine is degraded. Dopamine directly reuptaken by axon terminals, while glutamate and GABA use astrocytes to recycle. And yeah, this is the main idea. You should understand at least myelination, how myelination affects it, gated potentials, and all that jazz. So I guess I'll call it for today, and we can do more nervous system next time.